could divide it into these 4 chunks. So we do a lot of still method development for the fingerprinting methods. We also try to identify new molecules, and research-wise, we are interested into looking at the new mechanisms of disease. And also more and more, we are working with what people call personalized medicine, and I'm going to be calling more like precision medicine during the presentation, which is using all of the omics for diagnosis or also for choosing treatments. So this is a figure that is a bit more geared for our clinician colleagues, but it's just to show the possibilities of precision medicine. And I call it PANMOLECULAR in the way that is all molecules. So we are working with lipidomics and metabolomics, but of course, if we're going to have some genes that are going to increase our predictivity, we are going to be using them. So here we have the clinical data that the clinicians collect routinely. And if we can imagine this person as having a cardiovascular disease that in some years is going to affect their cognition, then we can say that by adding all of these omics, that as you see is an immense amount of information, and of course not the information that we have available to us just now, but I just put also some examples. So like genetics, it could be a polyform gene, which is a risk gene of Alzheimer's. But of course, there are many more genes, and so putting all of these molecules together, we can add to the precision treatment for this patient, or by adding a lot of biological signals on top of the clinical data. And of course, there are also environmental signals that we have to acquire if we can. So in the field of diabetes, the precision medicine strategies have been happening a lot in the last couple of years. And we have here the standard view of diabetes, in the sense that there is very prevalent diabetes, which is called type 2. And then we also have the immune type, which is type 1 diabetes, 2 other small ones. We are moving towards a lot of work into wanting to stratify patients with a bit more specificity. So there's been some work done on the clustering of patients, and by using 5 or 6 clinical metadata, then patients are classed into 5 groups. And then these groups are studied for, let's say, complications of diabetes. So what are their chances of maybe getting cardiovascular disease on top of diabetes, or neuropathy, or many other complications. And what we have been trying to work is more with this palette like strategy, where we wanted to look only in one person and then trying to define the phenotype of this person with all of the omics. And for this, we were working in a big consortium called Rhapsody, which actually had a lot of information and a lot of omics. So this just quickly our lipidomics method that we use. So for us, we have to run out of samples. So we have a bit of a generic method, although it's not shotgun, so it has a separation. And then we usually report anything from 300 to 450 repeats in 10 to 20 microliters of plasma. And so this is a figure that is quite busy, but I want to go through it because this is for us, or what I believe in our group in the hospital, what would be the ideal situation and a project. So what we would do is that we have 3 steps, and the first step is to have a look at all of these cohorts which we have analyzed already. And then ask one question, which could be who out of these 2,000 or 5,000 people actually happen to have kidney disease in 5 years. So we would get all of this data with the metabolomics and the lipidomics that we have run already. And then we would do some of machine learning to come up with a group of features, clinical and molecules, which actually describe these people for the complications. Then the second step would be to refine the analysis to targeted analysis, because at the end, we want this to be approved. And start screening in our hospital. And once we get a number of people with a high risk of that complication, then we go into step 3, which is a clinical trial. And all of these high-risk people are then randomized into a group which is active for the complication. Let's say this in this time cardiovascular disease, so you can give more aggressive treatment, or you can give a drug that is actually best for cardiovascular disease complications, or to do the standard treatment. And by doing the standard treatment, we will also find out if our panel is predictive. So we can find out the two things. So now I'm going to give examples quite quickly of things that we've done. We haven't been able to do the whole project or all of the steps, but we've done bits and pieces in each of the steps. So we're kind of gearing up to being able to do everything. So this is some of the panels that we came up in the past with the machine learning. And here were cases that were more difficult because the diagnosis is actually not very good or not even available. So first we were doing a lot of work with lipidomics and Alzheimer's. And then I'm going to give an example by Jin Xu, who's in my team at the King's College. And this was in a case where we were looking if we could predict the graft function of livers once before they've been transplanted. And this is a case where actually some molecules were actually more predictive than what they use for predicting the liver quality before the transplantation. But you see here some rock curves with like immense standard deviations, and it's because we didn't have that many samples. As you can imagine, it's very difficult to get the biopsies before a liver is going to be transplanted. And more into the diabetes field, this is work by Nawa that she was doing the PhD with us. And she wanted to find a panel to predict the future kidney complications for patients with type 1 diabetes. And she's come up with this panel, and it has the main features now that they diagnose actually for kidney function, which is as expected. And then we have a lot of sugar derivatives and ketones in this panel. And one nice thing that she did was to demonstrate how then we could do a calculation for one person and then give a prediction of if this person is at risk of developing a kidney disease in 5 years. And just as an example, this person will have a high risk of more than 80% to develop complications. So now this is now under review. And then this is also the step 2. So when we go and we do an assay, which is a quantitative, and as you all know, that's quite a lot of work. And I just want to give you an example on 3 cohorts that we ran. And these were quite big. Each of them is 1,500 people. And just to give you an example of one of the cohorts, which was this one in the Netherlands, also for type 2 diabetes this time, showed that one of these molecules was actually increase and more important in defining the cohort. And here we have a molecule called aminoadipic acid that there's already quite a lot of evidence that can be a biomarker of prediabetes. And as it starts also accumulating in the pancreas. And now we pass to the area of the clinical trials that we are very lucky that we have a lot of colleagues performing them. And this one in particular is one of alcohol intoxication. And it was done during a number of hours, and we had people with liver disease. So we had people that were healthy, they had alcohol liver disease and fibrosis, and also fatty liver disease. And this, I will just go through it very quickly. If people want to just see the paper in the Journal of Hepatology reports. We were very surprised doing lipidomics in this study, because what we saw in the healthy were quite a lot of things that were known, which is that the liver will require fatty acids, and then it will make a lot of triglycerides here in pink. But what we were not expecting is that the people with alcoholic liver disease will actually do the same as the healthy overall. And it was the ones with fatty liver disease where there were actually no triglycerides going back into the circulation. So we were not able to analyze biopsies from these people, but it looks like the lipids are actually retained in the liver, which is already fatty. So there is some sort of a loop going there. Then, okay. So then this is another clinical trial, and this is now a drug treatment with the liraglutide. And here we have 100 people with type 2 diabetes. So they were in two groups, and the ones that they were given liraglutide is a GLP-1 antagonist. And so when we had a look at the results of this clinical trial, if we were looking at the standard measures of lipids, none of them were changed with the treatment. So it wasn't only when we got the lipidomics results that we could actually extract what was happening with the liraglutide. And as you can see here, it actually decreases a lot of the triglycerides and some of the ceramides. And that was unknown. And also, we think that this might be one of the explanations on why liraglutide seems to be beneficial for future cardiovascular disease for patients with type 2 diabetes. And then Askar, what he did after that was to semi-quantify the famous ceramides that are associated with cardiovascular disease. And we had a look in another cohort. So we had a look in a cohort of 700 people with type 1 diabetes. And from those, around 100 actually had the cardiovascular events in 5 years. So this is really preliminary, and I just want to show you quickly. Here we have in the cohort, one of the ceramides, actually they are increased in this group that they had the cardiovascular events. And then we also had a look at the clinical trial with liraglutide, and we could also see that the ceramides were decreased with the treatment. So I am now in my conclusions. Lipidomics and or omics are key to precision medicine in diabetes. We think that we need a multi-step evidence method to actually assess clinical benefit that we can use in the hospital. Precision prognosis in diabetes will be linked to polypharmacy in the sense that we can find the combination of drugs that will work better for one person. Quantitative lipidomics and causal machine learning, I think, will catalyze the discovery of findings. Precision medicine, biomics, and machine learning will also feed information into the drug discovery pipeline. And to finish, I want to thank